Okay.
For presentation. Good morning, everybody. I'm representing the group of studies in moral learning development, GEDAN, the National Institute of Science and Technology in Molecular Medicine, NCTNM, both groups of the University Federal of Minas Gerais, UFMG. The title of my presentation is Association between the catechol o-methyltransferase, COMT, Val158 MET, polymorphism, and manual aiming control in health subjects. It's well known the role of dopaminergic neurotransmissions in cognitive processes, such as working memory, attention, decision-making, and inhibitory control. All these cognitive processes are associated with the prefrontal cortex functions. In the prefrontal cortex functions, there is an enzyme called the catechol o-methyltransferase, or COMT, that is responsible for more than 60% of the metabolic degradation of dopamine in the cerebral area. The COMT gene contains a functional polymorphism or mutation. This mutation is called Val158 MET. In the simple model, you can observe that MET individuals homozygous for MET allele has more prefrontal dopamine. The availability of dopamine is higher in individuals homozygous for MET allele than homozygous individuals for Val allele. Sorry. In general, the notion that the increased dopamine in the prefrontal cortex it will generally enhance functions is a oversimplification, because there is a relationship in prefrontal cortex states, the level of dopamine, for example, and the nature or demand of the task. Different from the studies that investigated the relationship between COMT and cognition, there are few studies investigating the relationship between COMT and moral control. Usually these studies have been used simple tasks and did not find significant associations in moral performance and COMT. Possible explanation is that the simple task seems to be more dependent on basal ganglia dopamine than the prefrontal cortex dopamine. So, tests that place more demanding on planning and monitoring should be used to investigate this relationship. An important area of prefrontal cortex is the dorsolateral prefrontal cortex. This area has extensive interconnections with regions directly involving motor functions, mainly premotor cortex and supplementary motor area. And the cognitive resources are associated with the dorsolateral prefrontal cortex and are recruited during manual motor control, which enable us to hold information and remember the desired goal of the task, to resist distraction and to stay on task, to resist responding too early, and to inhibit a prepotent and habitual response when the context is changing. So we designed this tool to investigate the association between the COMT polymorphism and motor performance in an aiming test that has different sensory motor requirements. Our general hypothesis is that individual homozygous for the MET allele would have better motor response than the individuals carrying a Val allele. Participating in the studies 111 participants, all right-handed, all cephalocyclic Caucasian Brazilians. The DNA of these participants was extracted from blood using the whole salt method. From the genotyping, we divide three different groups of participants with the allele MET, Val. Just to refresh, this Val allele is dominant, so we expect that MET, Val, and Val allele present the same type of phenotype, and the MET group that has more dopamine, a different type of phenotype. The paradigm tests were identical that used in previous studies, use a digitizing tablet, a laptop, and a software to control the stimuli and record data. Participants performed 100 trials. It's a boring task to maintain attention sustained in 4 different conditions. The control conditions, prepotent conditions, it was presented in 70% of the trials. The stimulus are presented on the screen. Subject put the pencil on the digitized tablet. This stimulus disappeared. In interval between 2 and 3 seconds, it appeared again, and subject and the participant need to move as quickly and accurately as possible. Another condition was the track condition. Instead to appear a green target, appeared a yellow target. The only difference was the color of the target. The aim was the same, make the movement as quickly and accurately as possible. Another condition was the inhibition condition. Instead to appear a green target, appeared a red target. This condition, the participant should to inhibit his or her response. And the last condition, for the higher ID condition, index of difficult condition, instead to appear a green target, appears a different target in terms of size and location. All subjects had 2 seconds to hit the target. After that, the test was closed, and we computed a spatial error because it did not hit the target. Our performance measures were reaction time, movement time, spatial accuracy, if the participant hit or not the target, inhibition errors, and kinematic measures were peak velocity, relative time to peak velocity, which measures related to pre-programmed phase of the movement, and number of discontinuities in acceleration in secondary sub-movement, are measures related to feedback processing. Analysis, we used two-way ANOVAs for analyze 3 genotype groups per 3 conditions in all dependent variables. Post-hoc test Duncan's, pre-square test for nominal dates, one-for-one correct for multiple comparisons, and we adopt an alpha level of 0.05 as significant. The main results in control condition, we observed that MAT group presented higher level of spatial accuracy than the VALMAT or VALVAL groups. That is, this group was more accurate than other groups. MAT group presented lower level of peak velocity than the other groups, a measure of pre-programmed phase. The structural condition, the MAT group presented a high level of spatial accuracy only than the MAT-VAL group, and MAT group presented a lower level of peak velocity than the other groups. To the higher ID condition, there is no significant difference among groups in terms of spatial accuracy, and MAT group presented a lower level of peak velocity than the other groups. There is no significant difference among groups to the inhibition condition. Overall, our hypothesis was confirmed because the MAT allele group presented better spatial accuracy with the same movement time of the other groups. Another pattern of results is that the VALMAT and VALVAL groups presented higher level of peak velocity than the MAT group. The first question is, why does the MAT homozygote produce lower level of peak velocity? Possible explanation is that an important variable in fast-ball direct movement is not only movement time, but also spatial accuracy. So, MAT allele carriers executed more controlled movement and thereby generating a lower magnitude of peak velocity during the trajectories. The main objective of this more controlled processing was to guarantee accuracy to the movement endpoints. Another question is why the better spatial accuracy exhibited by the MAT allele carriers was not observed under the high index of condition. The higher ID condition presents a different size and position of the target, Factors that require more flexible, more adaptive motor control from the performance when compared with the other conditions. In both healthy and psychiatric samples, individuals homozygous for MET allele show better performance in tests requiring stable performance than individuals carrying a whole allele. This group of results is specifically in cognitive tests. The same is not true when in cognitive tests when requiring more adaptive performance. So the relationship between dopamine and cognitive functions is non-linear and inverted U-shaped. The response is optimized within a range of dopamine activity that's associated with the test demand. In conclusion, these findings indicate there is a significant association between polymorphism and the manual M control in health subjects. This association was found specifically related to a component of movement preparation, the peak velocity. We did not find any association during the monitoring phase or related to feedback process. Furthermore, individuals homozygous for MET allele appear to be more accurate in the endpoint movement compared with the individuals carrying a whole allele. For the first time, that the influence of polymorphism on manual aiming control was observed, indicates that individual difference in motor control may to be investigated using genetic studies. Sorry about my English, and thank you for attention.
No need to apologize. Any questions? Quincy?
Thank you. Very well done presentation. I don't know if I missed it, but did you explain the reaction time results of it? You were measuring reaction time as well, right?
There is no difference in reaction time.
It's just
They represent only the main results. There's no. I think I will try to investigate in this line. I think that reaction time may be possible to be related with more basal ganglia dopamine. So the dopamine transporter, that maybe has a polymorphism that should be more associated with reaction time than the movement time.
Sorry.
No, go ahead. Go ahead.
It sort of strikes me that the entire task was essentially like a choice reaction time task where you have different options as to what you're going to be moving to each time, or if you're going to have to inhibit the movement. I would throw out one suggestion. If you have EMG equipment, I would love to see the premotor times and see what's actually happening in that inhibited condition, and if the people with the polymorphism are actually planning that or have the message out to the limb faster, I guess, than the other people. I also hope that your Brazilian soccer team has this polymorphism.
Okay. Thank you.
Thank you.